Give us a sense of the journey. Where did this begin and how did we get to nearly 36 patents?
Well, it began with collaborations with biomedical engineers. And the original motivation was really not to spin off companies. The motivation was to have an alternative route of getting funding for these research groups. So we apply for traditional grants, such as NIH, but they're very competitive. But we realized early on that we're also generating unique ideas. So as we had lab meetings, we would file patents, and then we would publish papers. And then when we got to a critical mass of patents, we could then spin out a company. And then often that company, when it raised funding, we could actually have our grad students work in those companies. So it became a very rewarding way to see our own mentees, basically to continue to work with them and to see them, have jobs and careers based on some of our ideas.
It sounds so simple. You have an idea, you get some mentees and collaborators, you get a little funding, and you've got 30 some odd patents. It's much more complicated than that.
Right. No, it's actually, I think the key is these brainstorming sessions. So we would meet weekly or bi-weekly in the School of Engineering at UT Austin. We'd have one or two professors present. We'd have graduate students present. We'd have myself and either medical students or trainees, medical residents present, sometimes cardiology fellows. And we would just have debates about ideas and just having clinicians and engineers together in the same room, the clinicians to point out the clinical problems, and then the engineers to suggest solutions on how to solve them. We would then basically come up with some unique ideas, and that's when we realized that we were generating IP, or the basis for patent applications.
Take us back to the first one. Now, there had to be a beginning, and what was that process with the very first patent and the first process?
Well, yeah, one of the first processes were working with Thomas Milner, a professor at UT Austin. We began using light to image mouse hearts just as a way to see the heart contract and relax. It's such a small heart, it's hard to do that with other technologies. And then we realized, we had the insight that maybe we could actually put that into a human coronary artery and see the same resolution, look for plaques that actually could rupture and cause heart attacks. And so that transition in that sort of insight allowed us to then focus no longer just on mice, but also then began getting access to human hearts and imaging human hearts and the plaques that were in those, and actually realizing that we could actually identify these thin cap fibroatheromas. The basis were when they rupture, they cause heart attacks. And up to that point, you could not image these thin cap fibroatheromas in live patients. They were only seen at autopsy, but it was known that they rupture. And so our group, and also a group in Boston at the same time, began imaging those in patients, and also ex vivo human hearts, and we began realizing that we could see those before they rupture, and maybe try to warn of future heart attacks for the first time.